My name is James Saenz, and right now I'm a group leader at the B-Cube Center for Molecular Bioengineering in Dresden. It's part of the Technical University of Dresden. And I have a lab that's interested in studying the role that lipids play in living membranes. And I did my postdoc previously with Kai Siemens, who Erdinch also worked with back in the old days. And before that, I did a PhD in chemical oceanography. Long story. I won't get into it today, but I've had a few different changes of direction over my career. But I've been in Dresden now for almost 10 years.
Yeah, great that you changed your course a bit, James. We are very happy that you're in the field of lipids now.
Me too, yeah.
So you can start sharing your screen, and okay. Whenever you're ready, go ahead.
Great, let me see if I can get this to work here. All right, can you see the presentation?
Looks very good.
Okay, great. So let me make sure I get this stuff out of the way here, and I'll start off. All right, so again, I'm really happy. Thanks again for letting me come in here and talk about our paper. The title of the paper that I'm going to tell you about today is Principles of Membrane Adaptation Revealed Through Environmentally Induced Bacterial Lipidome Remodeling. And this study falls under sort of a broader problem that our lab is trying to address, and that is understanding the design principles of living membranes, of biomembranes. And in particular, we're really interested in understanding what role lipids play in allowing membranes to do what they do. And one of the long-standing questions in the field has been, why do cells need such complex lipidomes? And we're interested in trying to see if maybe they can be simpler, and how simple, and what are the limitations surrounding that. Okay, so what I'll do is I'll give you kind of a brief introduction to motivate the whole thing, and then I'll tell you about the paper, and then I'll give a very short outlook at the end to give you a hint about where we're going next. So if we're interested in thinking about the design principles of membranes, let's start with a beautiful animation by Janet Iwasa of the membrane. And now this is based on the well-established fluid mosaic model. And what you have is a lipid bilayer. Those are the little lipids floating around there. And embedded in it are membrane proteins. And these are the things that do all the work in the membrane, from signaling to transport. And okay, so if we want to think now about the challenges that a cell has to meet in order to build a functional biomembrane, one is that it has to make sure that the material properties of the bilayer are optimized so that the proteins can function properly. And this means that it has to have the right viscosity and thickness and lateral tension profile so that these very complex protein machineries can operate properly. You can think of it analogously as having the right type of oil in your engine so that it runs properly. It's a bit more complicated than that. Okay, so the way the cell does that is it uses lipids to determine the material properties of the bilayer. So it has to balance the physical properties of the bilayer by determining which lipids end up in the membrane. A second challenge is illustrated by this animation that my colleague Ilya Leventhal made. And this shows a synthetic vesicle, and it's about to be pulled from the bottom there by molecular tweezers. And the main point of this is just to illustrate how mechanically stable membranes can be. And this is one of the key considerations that a cell has to make, is not only does it have to balance the material properties so that bioactivity can work, it also has to make sure that the membrane is mechanically stable enough to withstand all of the different conditions that the cell has to survive within. So if we want to start thinking about the most basic problems to understand in order to reveal the minimal design principles of a membrane so that we can understand them and maybe eventually even design them, these are the two things we have to think about. One is, how do cells choose the lipids that they put in their membrane, and the other is how do they make sure that the membrane is mechanically stable. Okay, so to put this another way, we know that living membranes, at least the ones that have been studied, which is fewer than we'd like, have constrained their material properties within a narrow range to make sure that bioactivity can operate properly. And so if we think now about fluidity is one example. Here we've depicted membrane fluidity from low to high on the left here. And okay, so living membranes are trying to keep that fluidity within a certain range. And the reason is, if the fluidity becomes too high, eventually the membrane falls apart and it no longer serves its role as a barrier. And if fluidity goes too low, the membrane becomes solid there's no longer any diffusivity, and of course, everything stops. So that's really important to establish these properties. Now, on top of that, it gets a bit more complicated because the cell has to achieve these optimal properties while everything's changing around it, the environmental conditions, physical and chemical. So let's imagine a scenario where we have a functional membrane, and we've depicted a very crude channel here, and the temperature decreases. Now, what happens is the lipids become more tightly compressed, something like this channel we've depicted might suddenly be shut closed because the lateral tension it experiences is too high. So the cell can sense that something's gone wrong, and it responds by introducing new lipids to the membrane to restore the optimal properties. And this underlines a key feature of membranes: they're responsive. So that's another thing that we need to really understand in order to get at the key design principles of membranes. Okay, now, last bit of introduction before we get to the data. How does this sort of simplified scheme that I've introduced now between fluidity and stability play out in terms of what life needs? We can imagine you start with a really simple lipid. It's saturated, it has short tails, it's very fluid but not very stable. We can increase chain length, and as we do so, we decrease fluidity but increase stability. Now let's think about this a bit. The lower part of this scheme is not tenable for life because it's fluid, but it won't produce a membrane that's stable enough to support a cell. The left-hand quadrant also isn't good for life because although it's stable, at some point when the acyl chain length gets too long, the membrane becomes solid and the cells die. So life wants to be over here somewhere. How do we get there? Well, there's a lot of different ways that cells use to achieve this. One, you can imagine you can add double bonds, and this allows you to fluidize the system without sacrificing stability. Or you can add a different kind of lipid like sterols or hopanoids. These can interact with saturated lipids to fluidize them. So in principle, it's very simple. All you need is a couple different structural modifications, and you can create a living membrane. But we know that living membranes are not that simple. They're actually really complicated in terms of the number of lipids that they have. So why? So think about it. Why don't cells make it this simple? Why do they have all these lipids? And second of all, if you think about all living organisms have very different lipidomes from each other. Bacteria are as different from each other as they are from eukaryotes. Is there some convergence in the types of lipid structures that all these different organisms have evolved to use? And can we explain that diversity with some common set of unifying principles? So those are some of the questions that keep me up at night. Okay. And finally, let's think about the complexity. We want to understand these principles of lipidome adaptation, but it's very difficult in mammalian organisms and eukaryotes, which have been the most well studied, because they have very complicated lipidomes, hundreds of lipids. It makes it very difficult to pinpoint specific patterns. Now, our approach has been to let's go to prokaryotes and think about some of the simpler organisms that might have simpler lipidomes. Now, there was a paper last year that showed that E. coli has just over 100 lipid species. So that's getting better, but still not as simple as we would ultimately hope to have a really nice experimental system. Then we can think about going really simple, synthetic cells, and maybe even thinking about ancient life and what sort of lipidomes might have been around when protocells emerged out of the primordial pools. Would it have been one lipid, three lipids? So these are interesting questions. So now, as we'll show, we'll discuss in the present study, we wanted to pick a bacteria that would allow us to see how a simple lipidome deals with adapting to environmental change. And to pick that organism, we had this thing growing in the lab, Methylobacterium extorquens, and sometimes serendipity plays a role. We had been studying that because it makes hopanoids. These are sterile analogs, and we were studying how they work in the membranes. But at some point we thought, well, let's take a look at this more carefully and see what the lipidome's doing. It's an interesting organism for this study because it lives on plant leaves, which means it's exposed to the outdoors. It has diurnal temperature changes of 20 degrees, changing light, chemical conditions, lots of things. So this is an organism that really has had to evolve to adapt. So it's a great system to see how membranes deal with adapting. So this study, basically, we grew the cells in the lab and we varied parameters like temperature, growth stage, salt. We shifted the carbon source from sugars to methanol, and then we looked at the effect of detergent on it. And now here's a graphical abstract, and it sort of gives an overview. So we started by growing the cells over varying conditions and harvesting them, and that's work that Sandra Risk and Daniel Grosser performed. And then we took the cells and we extracted them and measured the lipidome, and that's work that Michal Surma and Oksana Lavrinenko performed. We did this by shotgun mass spectrometry, which is a method that's well established in Dresden. And then Magda Ryczynska did a lot of work thinking about how to analyze that data and make some visualizations to see what kind of patterns were emerging. And Michal Surma also contributed to that work. And then we took some of these patterns and asked, well, what do they mean? What are the biophysical consequences of these variations? And that's where Zizek Twastek, a postdoc in the lab, came in, and he started to do these experiments in vitro to try to make some sense of the patterns we were observing. And finally, Helena Jambor is a data visualization expert who helped us to make the final visualizations, which also in turn helped us design some of the experiments. So there is a positive feedback loop between visualization and experimentation. Okay, so the first thing we did was just ask which conditions cause the lipidome to change. And one easy way, the simplest way to do that is we just took the data set and we analyzed it using principal component analysis. This is a method that allows you to take a multidimensional data set and simplify it down to 2 dimensions that explain most of the variability. And so on a plot like this, the distance between 2 points, each point is a replicate of a condition, and the input is the lipidomic data, so all of the lipid species. So the distance between 2 points here indicates how different the 2 lipidomes are from each other. So it's generally a measure of lipidomic variability. Okay, so what we see, the red dots here are the cells grown under the standard condition, 30 degrees Celsius, pH 7, defined media. And you see a lot of the points cluster very closely to it, indicating that most of the changes we introduced didn't change the lipidome that much. But there are 3 conditions that did have a relatively large effect on the lipidome. One was higher salt, when the cells went into stationary phase, when they stopped growing, and also temperature. And temperature had a very large effect, especially at these very extreme temperatures, like 6 degrees. Okay, so we know that some conditions change the lipidome, others don't. And temperature in particular, for the range that we tested, had a very large effect. So we'll come back to that later. Now we wanted to ask, okay, so what lipids were responsible for these variations and how many were actually varying? So to do that, we have to actually look at some data. So what you're looking at here is a table of all of the lipids that we measured. So there's the phospholipids and there's a couple hopanoids down here. This covers about 90 mole percent of the, over 90 mole percent of the lipidome of Methylobacterium. What we're missing are lipopolysaccharides and a few of the minor hopanoid species. Okay, so the way to read this table, on the right here, we have each row is a lipid species, and the dots show the abundance of that lipid species for a particular replicate for all the different conditions. And the red dots indicate the standard growth condition. Okay, so there's this part, which is the actual data. And then we've grouped this table into the different structural features that the lipids have that are important for dictating the physical properties of the membrane. So the first thing we can see on the left is head group. So there are 4 different head groups in the phospholipidome that we measured. And just to give a brief introduction for anyone that may not be super familiar with lipids. So the lipid head group is, the core component is this phosphate attached to a glycerol, and then there's 2 acyl chains that come off of that. So that's the thing that they all have in common. Now, we can look at how they differ. So you have phosphatidoglycerol has a glycerol group stuck to the head group. Phosphatidylethanolamine has an ethanolamine. We have choline, and then we have cardiolipin, which is basically 2 glycerols sticking off. So these of course can influence membrane properties by changing them. Secondly, we have the acyl chain length. So what does this mean? We're talking about the cumulative chain length of both acyl chains. So in Methylobacterium, we have 16 carbon chains and 18 carbon chains. So that means we can have 3 different cumulative chain lengths of 32, 34, and 36. Those are listed here. Then we have double bonds, and these are very important for modulating membrane fluidity. And in Methylobacterium, we showed that there's double bonds at a delta 11 and delta 5 position. The delta 11 position is the most abundant. This means you can have lipids with up to 4 double bonds per lipid. And we've listed those all here. Okay, so now you've been introduced to the lipidome. One important point is it's very simple. We only detected 27 lipid species here, 25 phospholipids, 2 hopanoids. So this is putting us in the lower part of that plot I showed previously. Now we want to find a way to quantify lipid species variability so we can ask how many lipids are actually changing when we alter these parameters. Okay, so one way of doing that is we just quantify the range of variability for each lipid species. And you can do that either by, for example, taking the standard deviation of a range of abundances or just measuring the max and min abundance. And both of those approaches yield qualitatively similar results. Now we can go and we can replot this data. So what you're looking at here is the same data, but now we've sorted the lipids from highest to lowest based on their mean abundance. And these gray bars show the range of abundances for each lipid species. Okay, so now what we can do is, for example, for this lipid, PE362, this is shorthand for the head group chain length double bonds. We can, for 2 different conditions, we can evaluate the standard deviation of the range of abundances for that condition. And that gives us sort of a measure of lipid variability for that species for 2 different conditions. And then we can take those standard deviation values and plot them as indicating variation. And we made a heat map here with the scale going from yellow to red. Okay, so here we go. Here is all the lipid species for all the conditions that we tested individually. And the first thing that jumps out is not that many lipids are actually varying very much. Most of these boxes are yellow, meaning that there's very low variability. Okay, another thing that jumps out is most of the highly variable lipids are the most abundant. But with a few exceptions, we see that these polyunsaturated lipids here are highly variable when temperature changes, and this makes sense, and we'll get to this in a bit. Okay, so now we asked how many lipids are actually varying for any of these conditions, and to do that, we basically asked how many lipids account for 90% of the total variability. And when we do that calculation, we get somewhere between 11 and 13 lipids are highly variable for any of these conditions. So that's an interesting number because it provides an upper constraint on the maximum number of variable lipids you'd need to build an adaptive membrane. It may be less than that, but this is one data point that we have. We can say, okay, you need to know how many lipids you're going to need to build your synthetic membrane to make sure it can adapt to all these conditions. You'll need 11. Okay, but which lipids? That's another important question. What's the logic behind how the cell chooses which lipids it varies? Okay, so to do that, we go back to our table here, and we can think of another way to analyze the data set. We know that the structural features of the lipids are important for dictating the material properties of the membrane. So what we can do is reanalyze this data set by grouping lipids with similar structural features. So one way is we can group the lipids based on global features. For example, the head group. We can add up all of the lipids that have a certain head group like PG, PE, and PC. And then we can say, all right, what's the relative abundance of these different head groups and how do they vary? Another way we can do this is we can ask, we can group all of the lipids with a certain number of double bonds. So we can say all the lipids with 2 double bonds, we add those up. And then we do that the same for lipids with 1, 2, and 3 and 4 double bonds. And then we ask, how does the relative abundance of lipids with different number of double bonds change with the conditions? Okay, another thing we can do is go a little bit more into detail. And instead of grouping things globally, we can say group the acyl chain features within each head group class. So we can ask within PG, how do the relative abundances of lipids with different numbers of double bonds change? And this gives us an extra detailed perspective on what some of the patterns might be that the cell is using to adjust its lipidome. Okay, so let's take a look now. We'll go to temperature because we already established that that was the thing that caused the largest variation. So first we're looking at class. We're looking at the abundance of the different head groups and the diplopterols, the hopanoids. And what you can see, we look at the abundant mole % abundance or from temperatures from 6 to 30 degrees Celsius. And over most of this range, there's not much going on. When we get down to 6 degrees, a lot of stuff happens. PC and PE sort of switch places. But this is a very extremely low temperature. For most of the sort of normal temperatures that this cell would experience in a typical diurnal cycle, there's not much going on at the class level. Now let's look at chain length. Again, we see something similar. There's a big change at low temperature, but for most of the temperatures, it's essentially constant. So not much to see here. Now double bonds. Now we're starting to see something. Okay, so what we're seeing is they're changing. So that's good. And how are they changing? Well, this is sort of what we'd expect based on what's known about homeoviscous adaptation. It's well established in bacteria and also eukaryotes that when temperature changes, the cell compensates for the fluidity change induced by the temperature change by altering the number of double bonds per lipid. And it adds more double bonds as temperature decreases. And that's exactly what we see. We see that the lipids with 1 and 2 double bonds decrease at lower temperatures, and the lipids with 3 and 4 double bonds increase. So that's good, that makes sense, but it's not necessarily surprising by any means. Okay, so let's look at a little bit more detail. Now, instead of looking at the global features, so we're looking at the total change in double bonds per lipid, let's now look at how that changes within each head group. Okay, so now you see a figure here, which is a giant mess, but bear with me for a second. So if we look in the middle here, these two lines, these two olive green lines, are PC lipids with 2 and 3 double bonds. And we can see that they vary tremendously. They basically switch places. We go from having predominantly 2 double bond PC to predominantly 3 double bond PC. This is a huge contrast to PG, where we see almost no change at all. So the bulk of the unsaturation variability with temperature is due to PC and PE here, right? So that's kind of interesting. We didn't expect that. And it's not something that's been widely documented in bacteria for sure. Okay, so let's look at this a bit more. Let's step back a bit and then see if that holds for all the conditions. So now we're looking at the same kind of way of plotting the same way of processing the data plotted in a different way. We're looking at the number of double bonds per lipid within each head group for all conditions. So we're looking at the range of abundances for all the conditions. And of course, you can right away see that PC here has the largest spread and PG has the smallest. We can quantify that, again, by looking at the standard deviation of these values as a measure of variance, a measure of variability. And okay, so now we're plotting the standard deviation of these values for each head group for 1, 2, 3, and 4 double bonds. And it's very clear. PC, PE are highly variable, PG is not very variable. So this really holds for all of the conditions that we tried. There's a non-even distribution of acyl chain remodeling between head groups. Okay, then we asked, is this regulated somehow? Does this change over different intervals of, say, temperature? So here we've plotted something similar but slightly different. We're plotting the contribution of each head group to the total change in unsaturation for a particular temperature interval. So in this case, we're looking at the difference between 30 and 20 degrees Celsius. And we see PC and PE are the most, contribute the most to unsaturation variability. Now we go down 20 to 13, PE becomes more dominant. So that's interesting. Then we go down 13 to 6. Now PC is the most dominant. So you can see that this is not something that's a fixed ratio. It's changing with different temperature intervals, but suggesting that this might actually be regulated and there might be some logic behind how they're deciding where to modify acyl chains, depending on the condition, the temperature range, for example. Okay, so another question that comes out of this is this something that's just a quirk of this weird bacteria that grows on plants, or is this maybe, are there broader implications for this kind of head group specific acyl chain remodeling? Okay, so to answer that question, we looked to a previous study that characterized lipidome adaptation in yeast by Klosa and Miha Surma et al. In PLOS One back in 2011. Now, first, we've replotted the data here, and we're looking at is for the whole range of temperatures, the contribution of each head group to unsaturation. And again, PE and PC are the most variable. Now, when we recalculate the data from Klosa et al., now I apologize, the citation isn't here, but this is data from Klosa et al. In PLOS One back in 2011. If everyone's curious, I'll post that on the YouTube channel. So they put all their raw data online, and we reanalyzed it. And this yeast has more lipid head groups, of course, it's more complex, but we've highlighted PC and PE here, which they have in common with Methylobacterium. And we again see that PC and PE are the most variable, like we see in Methylobacterium. So again, this is now appears to be something that's also present in eukaryotes, at least in yeast, and may well be a sort of pattern of lipidome remodeling that's universal, or at least widespread. But that still needs to be determined through more studies. Okay, but this then, the final thing I'll tell you about we then asked, well, okay, so what? What consequence does this have for the material properties of the membrane? Is there something significant about varying acyl chains in one head group versus another for the effect that's made on the membrane properties? Okay, so that led us to design an experiment where we made liposomes, so synthetic membrane vesicles, with 3 different lipid classes, PC, PE, and PG. And each lipid class was made up of 2 lipid species. One species had palmitate oleate, and the other species had 2 oleates. So one lipid had 1 double bond, and the other had 2 double bonds. And those are abbreviated here as PO and DO. So we started with a control mixture, which had a molar ratio of 1 to 4 of these 2 lipid species for each head group the same. So each head group has the same ratio of these 2 different lipid species. Then we changed all of the ratio of those lipid species the same in each head group, so that the final acyl chain composition, the final composition was 2 to 3 of PO to DO. So we increased the number of lipids with 1 double bond, which of course should decrease fluidity. Okay, so then we get this final total acyl chain total ratio 2 to 3. Then we tried something different. We tried only changing the acyl chain distribution in one group while keeping it the same in the others. Okay, so but in a way that we still get the same final total acyl chain distribution as we had in this condition here. So we did that for PC, PE, and PG. We just overcompensated in the changing the ratio here without changing the ratio here, so that we'd still get a 2 to 3 total ratio. Okay, I hope you get that. I had a lot of trouble explaining this to people. It's not super straightforward, but hopefully you get the point. Okay, so then what we did is we measured these liposomes. We measured a parameter called, we used C-lardin, which is a fluorescent probe that is sensitive more or less to lipid packing in the membrane. And we use that because this is a parameter that's very sensitive and is affected by a lot of different physical properties of the membrane. So it's a robust global readout of changes in the membrane's physical state. Okay, and we measure lardin, we get a value called the general polarization index, which can range from 0.5 to minus 0.5 practically. And positive values are less fluid and negative values are more fluid. Okay, so our control mixture here, starting point, has a value of about minus 0.3. Then we increase the amount of lipids with 1 double bond, and sure enough, the GP values increase, meaning that the fluidity decreased. That's what we expect. Okay, then we try our experiment. Now we only vary the acyl chains in PC, and we get the same result, so nothing surprising there. Okay, but then moving a step forward, we tried this for PE, and that's where we got a surprise. There we see that the effect on lipid packing is much larger than just varying in PC or varying in all the lipids the same. So it actually matters which head group you choose to remodel your acyl chains in. When we tried this with PG, we got a different result. It's actually less sensitive, so it goes the other direction. So this was a surprise. To our knowledge, this hadn't really been looked at before. And it now suggests that these unique patterns of head group specific acyl chain remodeling that we observe in Methylobacterium and that are also present in yeast may have a biophysical consequence, and this may be a mechanism that cells use to fine-tune their homeoviscous adaptation response. And importantly, what this shows is that even a simple lipidome, by varying the acyl chain remodeling within different head groups, is capable of increasing the degrees of freedom it has for lipidome adaptation, right? So this adds a whole new level of variability that a lipidome can enact in order to adapt, that we have to take into account in thinking about mechanisms for lipidome regulation. For example, what are the, we have the biochemical pathways for all these lipid synthesis pathways. Where do we account for the substrate specificity that would be required to achieve this sort of head group specific remodeling? I think that's a very interesting question that hopefully someone figures out at some point. Okay, so to summarize, we've examined the lipidome of Methylobacterium extracans. We've shown that it has a very simple lipidome, and we've shown that maximally at least 11 lipids out of the 27 total lipids that we measured are involved in variation. So that provides an upper limit on the number of lipids that you need for an adaptive membrane. We showed that remodeling of acyl chains is unevenly distributed across all lipid classes, and this has biophysical implications. You get a different effect depending on which head group you remodel. And we propose that this may be a novel mechanism for fine-tuning membrane adaptation. Okay, now very briefly in the last couple of minutes, I think we're more or less on time. I just want to give an outlook on where we're headed with all this. This was the first step for the lab to get at some of these questions we're really interested in. Now we're trying to figure out a way to get more control over the system. So we've identified a new model organism, mycoplasma, and its synthetic derivative, the minimal cell, which the Craig Venter Institute has developed, this organism called JCVI SYN 3.0. These organisms are very interesting for us because, first of all, they're some of the simplest organisms that you can grow in the lab. The minimal cell has fewer than 500 genes. They have one membrane and no cell wall. So you're basically looking at a liposome with a cytoplasm. So experimentally, terrific. Now, the other nice thing about them is they don't synthesize their own lipids. That means that we can actually control their lipidome just by dictating which lipids we give them in the media that we grow them in. So this affords us an unprecedented ability to now explore the relationship between the complexity and composition of the lipidome and the function of the membrane and ultimately cellular viability and the ability to adapt to different niche environments. Okay, so going back to our scheme here, we have complexity of lipids, and we've now shown that methylobacterium puts us at the lower end of this spectrum of prokaryotes. Now, can we go simpler? And our hope is that with the minimal cell, we can start to approach one lipid, or at least get near it, right? And we can begin to ask questions. What happens as you begin to get close to this critical threshold of the minimal number of lipids required to support a biomembrane? What sort of functions are limited? How does the cell's ability to adapt to conditions change? And this will start to allow us to address questions, for example, how, if we wanted to engineer synthetic membranes for synthetic cells, what would we need in terms of the lipid repertoire? And also, what was involved at the origin of life? How many lipids would have been required to assemble a membrane that might have given some advantage to an emerging, say, an RNA world or whatever was happening back then? What was the protocell lipidome potentially like? So one thing, in addition to looking at those lipidome complexities, we also need to look at the output, which one of them is the material properties of the membrane. Now, here's some data that a student, Natasha Safronova in the lab, has done on the mycoplasma grown on a complex lipid diet. And we've just measured the membrane microviscosity using DPH anisotropy over a range of growth temperatures. And we see that there's a range where it's able to maintain its viscosity. And then if you go above or below that, the system breaks down. So what we're trying to explore now is when we vary the complexity of the lipidome, how does that affect the cell's ability to maintain its viscosity? And how does it affect the range that can still support life? And when does the system break down and cause the cell to die? Now, and this is all work that Natasha Safronova is doing for her PhD thesis, and hopefully we'll have a beautiful manuscript on bioRxiv soon. But finally, this is not a one-dimensional system. I talked about viscosity a second ago. But the membrane, there are many important properties. For example, phase criticality, the ability for the membrane to modulate its phase properties, for example, potentially for rafts, to laterally organize components. Permeability is also important. There are many other important parameters. So we can now imagine a multidimensional space of physical properties that the cell has to find its path through to optimize in order to achieve to be alive. So we can imagine there's this volume in a multidimensional space that the membrane has to optimize and it has to stay within. And of course, the volume probably isn't as boring as this cube here. It's probably, something more like that. So one hope is that we can begin to explore the shape and the volume of this space, this multidimensional space, using the mycoplasma minimal cell systems that we're developing, and begin to understand, what are the parameters that differentiate a living membrane from a dead membrane. Okay, so with that, thank you very much. Of course, I have to thank my whole lab. There's a picture of us performing safe social distancing measures. Of course, thanks to the Technical University of Dresden and the B-Cube, which is my host institute, the Volkswagen Stiftung for funding our life initiative with Ilya Leventhal, Robert Ernst, and Maya Schuldener, and as well as the BMBF for very generous funding to support our lab. Many people contributed generously to discussions and giving us feedback, and I'm always grateful to that sort of kind, open sharing that's really a hallmark of the Dresden community that I've experienced. And also particular thanks to Lipotype for all of their help, and as well GCMS analysis were carried out by the DSMZ in Braunschweig, headed by Dr. Meine Neumann-Schall. So with that, I'm very thankful for the opportunity to speak, and I'll take your questions.
Thanks a lot, Jimsy. Amazing. Really, really great talk. I'll start with a question myself. So you present this homeostasis, which is now clear that it happens in eukaryotes, it happens in prokaryotes, and you show that it basically the bacteria try to keep the fluidity the same. The question is why? Which property, which output actually they want to keep constant? Is it the protein, the diffusion of proteins on the membrane? Or is it just basically their capacity to move? What do they want to keep constant?
That's a great question, and that's exactly one of the things that we're really interested in trying to understand. There's a lot of focus on viscosity, and we don't necessarily know that that's the most important parameter. In fact, we don't really know what are all of the important parameters that we need to be paying attention to, let alone what we can actually measure. But just a brief example, there was a very nice paper by Ite Budin last year in Saenz where they showed that they can artificially vary membrane viscosity in E. coli. And one of the key things that I took out of that paper was that the viscosity seemed to be a very important parameter when the cells were performing respiration. And the hypothesis in that paper was that this is because of the diffusion of membrane-associated electron transporters like quinones as a bottleneck. But when the cells are doing glycolysis, when there is no diffusive bottleneck in the membrane, viscosity becomes much less important. Henrik Strahl is also generating interesting insights into this. So it looks like viscosity may not always be that important. In fact, cells might be able to tolerate a pretty broad range of viscosities. But something must be important, because when we measure the lipidome of these organisms, we see that they control it. Even mycoplasma in the minimal cell have a tight control over their lipidome when we change parameters. So they're putting a lot of effort into controlling which lipids end up in the membrane. So there must be a good reason, and it probably has something to do, at least partly, with the physical properties of the membrane. But we still have to figure out what are the right properties.
Thanks a lot, James. So I will start with the questions by the audience. We have a lot of questions. I hope you still have some time. So first question is by Ed Lyman, and he is asking his nasty question. Is there a one-to-one correspondence between Lorden GP and fluidity, or might Lorden GP alone be insufficient to determine changes in fluidity?
Well, yeah, no, I wouldn't use Lorden GP as a measure of membrane fluidity at all. I mean, what it's really telling you is how much water penetrates the bilayer, or what position the Lorden probe is in the bilayer. That can vary as well. So yeah, I mean, there's been a number of studies. Erdős has done a lot of work showing how parameters like GP can be correlated with fluidity or viscosity. And in some cases it is tightly correlated, but in other cases it isn't. So no, I wouldn't rely on that. But it is a nice, easy, robust readout if you want a quick idea of whether something's changing in the membrane.
Related to this, James, did you use any other probe? Because you showed that with C-Lorden GP, lipid head groups might be differently sensitive. Is it because they are differently sensitive? Is it because they are globally different in changing the lipid viscosity, the lipid packing? Or is C-Lorden more sensitive to one of the lipids compared to others?
Good question. So my postdoc, Zizek, who led a lot of this work, had that very question, and we checked just using Lorden instead of C-Lorden, since that doesn't have that extra bit, which may interact with PE, for example, and we got the same result. So it wasn't an artifact, for example, of some sort of electrostatic interaction. But we haven't tried other things. I mean, it would be really cool to actually dig into in more detail, what are the physical differences that are happening here.
Sounds great. Okay, James, next question is by Mathieu Chauvin. I guess membrane proteins are present in these membranes. Can you comment on the evolution of the lipid composition for membrane proteins?
Yeah, absolutely wonderful question. No, I can't comment on it, but it's a really good question. I mean, there must be some coevolution of the transmembrane domains of proteins with the specific lipidomes of these varying organisms. I mean, that's not something we have gotten into yet, but there's sure to be some really interesting things. I mean, like Ilya Leventhal did some nice work looking at the features of the transmembrane domains that are linked with association with, different ordered or less ordered domains, RAF domains. So that sort of analysis may, clearly will provide insight into whether there are really big modifications to transmembrane domains based on lipidome evolution. Super interesting. No, we haven't dug into that yet.
Great. So next question is by Alexander Jokens. Could it be that the enzymes in charge of adding double bonds only recognize PC as substrate? That is why at low temperature, there is a high increment in PC. And I'm adding on top of this question, if there are different enzymes involved in adding double bonds in the metabolism, right? Maybe some enzymes are not active at low temperature, while some, like the ones that are recognizing PC as substrate, would still be active. So that's why you would see the differences.
Yeah.
Do we know these enzymes, first of all?
No, well, kind of. Two parts of that answer. Yeah, so as far as we know so far, the double bonds in the acyl chains are derived entirely during the process of acyl chain elongation and synthesis, before the synthesis of the lipid. So they're fed into the phospholipid pathway. We do not know if there are lipid desaturases that can act on lipids in the membrane, as is the case, for example, with bacillus, with the DES system. But there may well be. I mean, this bacteria has not been exhaustively characterized. So I think that's an open question right now. What are the mechanisms of unsaturation, acyl chain modification in general, and how can we account for this sort of variability? Yes, maybe temperature itself is just a limiting factor. And that raises another problem or question even, which is how do we know that all these variations in the lipidome are really controlled and not thermodynamically driven by temperature and other parameters? And I think especially, as we move forward, that's going to be an interesting problem to really nail down. Is this really on purpose, or is it just, thermodynamics and accidents?
Thanks, Jimsy. Next question is by Maria Makarova. Can lipid head group shield the membrane and affect how much of water molecules will get into it, and therefore influence load on reads?
Yeah, I think, I mean, if you now, I don't have any specific example there, but I would imagine yes, that the chemistry of the polarity of the head group would affect how much water can penetrate, how tightly packed the head groups are. That's where you have to be really careful with Lardin. If you're looking at changes within a membrane that has a similar head group composition, then you're probably pretty safe. As soon as you start to change the head group or introduce weird lipids like archaeolipids, then you have no idea if the amount of water penetrating is changing or the position of the probe is changing or whatever. So that's where you have to really use some caution.
We should measure diffusion, James.
Yes, yeah, I have to get out there to Sweden. Okay,
next question is by Jun Kawamoto. Interesting. Would you explain about the head group specific acyl chain remodeling in bacteria? This is related to question before. Does bacteria have acyl chain transferases recognizing head groups of phospholipids? You partially said you don't know, but maybe you have additional comments.
Well, in Methylobacterium, we absolutely don't know. And I think that's really an open question still. In Mycoplasma, we've actually made a lot of headway in beginning to sort that out. And we've shown now that Mycoplasma do have acyl transferases and lipases that allow them to remodel the lipids we feed them. So they can swap acyl chains around. And that's, well, it's a bit problematic for us in some ways, but we're now working to identify what are those genes actually so that we can knock them out. Yeah, so some bacteria do, but in this case, Methylobacterium, we're just not sure.
Thanks, James. So next questions, actually, two of the audience is asking the questions towards same direction, Betula and Christina. And the question is, can various organelles such as mitochondria adapt to changes in the environment by altering their lipid composition by similar mechanism, if you think about the endosymbiosis especially? And Christina is asking in the same line, does functional imaging of organelles such as mitochondria and ER that elongate fatty acids help in understanding membrane fluidity?
Yeah, I mean, definitely not my area of expertise getting into the eukaryotic cell, but what my understanding is that this is an area that has not been very well explored. I mean, partly it's very tricky to purify the organelles to get reliable enough lipidomic data. There has been some work. Robert Ernst recently did this on the ER, I believe, in yeast, and had some interesting studies, results there. And it makes complete sense that the organelles also need to manage their lipidome profile. There are different challenges there, of course, like with the mitochondria, they get some of their lipids through these contact sites from the ER. And so there's a different mechanism there of how they would regulate the lipid input. Actually, in some ways, it's analogous to the problem of mycoplasma, my new favorite organism, because they also have to get their lipids from the host organism through some kind of contact with the cell. So yeah, the organelles most likely do that, but I don't think this is a exhaustively studied area yet.
Brilliant. These are young people, so they're now hearing out what is the next steps to come for them.
Exactly.
So the next question is also by Christina and Edge, and they are asking from different angles, but again, it's similar line. So the question is, do the proteins, for instance, forming the ion channels in the membrane, regulate the cell fluidity? Would proteins contribute to it? And the next question by Edge is saying, could oxidative stress synergize with osmotic stress of the membrane in remodeling of lipidome?
So the first question is about whether proteins can induce changes in the membrane. I think that's a really interesting area that has not received much attention. I mean, there is a paper by Kaiser et al. From the Simmons lab back in 2011, where they showed that synthetic peptides have an ordering effect on lipid bilayers. So in that case, yes, introducing a peptide to the membrane can increase its order. I believe there's some work in Bacillus that it will be emerging or has emerged soon, showing that some of these proteins associated with the membrane domains may also have an effect on membrane properties. So yes, that's a really interesting area. It is another area that is not well explored. And it's very likely that proteins can modulate membrane properties and that cells take advantage of that. What was the second question? Was
if the oxidative stress would contribute to these changes?
Yeah, I would imagine. I mean, that you're introducing changes in the rate of degradation of certain lipids, so that, unsaturations and stuff. So that would, of course, provide a challenge to maintain a steady state in a growing system. I'm not sure how otherwise free radicals directly affect the physical properties of the membrane, but maybe that's also they're in such low concentrations. It's probably not a major factor. But yeah.
Yet another unknown, James. So next question is by Pablo Caravilla. Very nice talk and approach to the problem. Congratulations. PC and PE neutral show higher variability as opposed to PG and cardiolipin negatively charged in response to the changes. So according to your liposome results, the head group has a big impact on the fluidity of the membrane. Would you have an idea why this is? The lipid shape, charge? What could that be?
Yeah, very good question. So remember, we're keeping the ratio of the head groups constant. So the effect we're seeing is not due to, of course, varying the ratio of the head groups. But yes, so why is there such a difference? I mean, in PE, you could imagine, first of all, that it has a different geometrical profile from PC. It has this a more conical profile. And so changes in the freedom of motion of the acyl chains, in that sense, may have a much more dramatic effect on the conical shape of the lipid. So maybe that's one reason why PE might induce more of a change. The other is that, of course, PE can engage in hydrogen bonding, and so perhaps the distance between the lipids caused by changing in the number of double bonds is also affecting the degree to which the PE can hydrogen bond with neighboring lipids, and this might be another factor that's introduced. But that's, those are just speculation at this point. I think it's a really interesting area to dig more into by a real biophysicist.
Okay, James. So the next question is by Jansu. She's asking, as you stated in the example of PC and PE, the adaptations differ from each with temperature. Would it be possible to find the optimum temperature for the lipidome by examining factors such as diffusion and nutrition, in addition to the growth that people usually measure?
An optimal, in other words, to vary the external conditions rather than varying the lipidome, vary the external conditions to optimize. Yeah, very good question. We've been trying that with mycoplasma, and so far we haven't had much luck. But the idea is, that we would try to vary pH or calcium or temperature to tune the membrane properties with a constant lipid input and modulate growth. We're still working on that, but that is something that's now possible.
James, next question is by Yu Qin. Great talk. Cardiolipin has 2 head groups and 4 acyl chains, yet it is so stable and insensitive to temperature change. How come?
Good question. It may be that whatever it's doing is important for other reasons besides homeoviscous adaptation. So the relative charge distribution, for example, or there's some work suggesting that maybe a cardiolipin is important for tightly curved regions at the poles of the cell. So maybe just maintaining the intrinsic curvature at those regions is important, and that's why cardiolipin isn't varied much. I don't have a concrete answer to that, but it definitely raises the point that the invariant lipids are as important as the variable lipids in the sense that they must be doing something very important if they're that conserved. But we need to figure that out.
Okay, so next I'm going to combine 2 questions again. So next 2 questions are by Iram and Hussein, and it's more about general lipidome changes. What can this lipidome change in physiology of the microorganism? So Hussein is asking, cancer cells divide constantly, and this uncontrolled proliferation be triggered by change in lipidome. So it's prokaryotic cancer cells, but can you adapt it to more prokaryotic system? Would it change the cell division rate, for instance, in bacteria?
Yeah, I mean, again, this is something we haven't controlled or tested, but well, okay, we have done experiments in the bioreactor where we grow them in a chemostat and we can vary the growth rate by changing the dilution rate of the reactor. And that induces a change in the lipidome, not huge, but moderate. So changing growth rate can change the lipidome. Conversely, I mean, if you imagine that the ability to divide and grow and build new membrane is dependent on a lot of proteins working optimally, then yes, maintaining physical properties that are optimal for those proteins function would have an effect on the rate at which the membrane can form itself. That's the most direct link I could think between lipid remodeling and cellular growth rate. And
Iram is asking in the opposite direction. Would it contribute to cell death?
You mean
lipidome remodeling? Would it somehow change the signaling of cell death?
Yeah, I mean, you could imagine, first of all, if you don't have an optimal lipidome, the range of conditions that the cell can tolerate is shifted or reduced, right? So that's one thing, that you might be right at the edge of death, and then something goes wrong in your lipidome and that pushes you over. So I think for sure this is an important parameter.
Okay, James. So last comment by Jun Kawamato, who asked a question and saying, thank you very much for your presentation and answering my question. I'm seeing you from Japan.
Wow, you stayed up that late.
Yeah, and I'm also interested in acyl chain remodeling enzymes working at extremely low temperature. So I suggest that probably he or she will get in contact with you if that is the common interest.
Yeah.
So I have many questions, that my own questions, but I'm not going to ask because we are coming to the end. But I will ask one single question as the last question. So you said 11 lipids out of 27 you tested are somehow going up and down. What about this other 16? What are they doing? Why do we have so many lipids, James?
God, why do we have so many lipids, Erdinch? I don't know. Well, most of those lipids that are not varying are also very low abundance. So these are things that are less than 1 mol%. So they may just be, well, I really doubt that the cell is so disorganized that it's letting stuff just sit around. They must be serving a purpose. There are other things besides just physical adaptation of the membrane. Of course, lipids play a role in interacting with proteins and signaling. I mean, all of these low abundance invariant lipids may be playing some conserved role in providing a microenvironment for certain proteins, or for providing a sort of a constant background. No idea, but I think that's, we focus on everything that's varying, but the stuff that's not varying, well, we're also going to need that to build membranes. So we should probably give it some attention.
And James, it was very surprising to me that you didn't talk about hopanoids.
I didn't.
This changes in hopanoids, you didn't even show. How did they change?
I didn't even show a hopanoid. Yeah, they do change. I mean, there's actually some really interesting results in there that we didn't dig into in this paper even, but you know, modifications. So hopanoids are sterile analogs that we've been studying, and they do similar stuff to sterols in these bacteria. But this bacteria makes multiple hopanoid structures, and they seem to be changing those structures depending on temperature and other parameters. So yeah, hopanoids are doing some interesting stuff. I'm actually writing a DFG proposal now to get more money so we can dig into these questions about why the bacteria make so many hopanoids and why are they adjusting them and what's the purpose. But maybe we can talk about that for the next session.
Yeah, I can already see the title, hopanoids.
Hopanoids, exactly.
Exclamation mark. So James, that was brilliant. Thanks so much. There are a lot of comments about how nice your presentation was.
Well, thank you very much, and thanks to people that's in the East that stayed up so late. I really admire the ability to stay up so late.
I'm sure it was worth it for them. Thanks so much for this great talk, and we keep in touch.
Great. Thanks for having me, Ardent. It was a real pleasure. And anyone who has questions, feel free to email me or just get in touch. So.
Thank you, James. Thanks a lot.